Hi, I'm Cole the Christmas Angel. I'm here today to show you how an amino acid chain or protein is formed from a specific DNA antisense strand. Let's start with this DNA antisense strand. TAC, AAA, CCC, GAG, CAT, ACT. When an antisense strand is created, a sense strand is also created. In this case, the sense strand is ATG, TTT, GGG, CTC, GTA, TGA. As you can see here, the sense strand is the exact opposite of the antisense strand. The next thing that happens is the synthesis of messenger RNA, also called mRNA, from a DNA template. So here's what happens. First, DNA unwinds. Second, RNA polymerase recognizes a specific base sequence in DNA called a promoter and binds to it. It identifies the start of a gene, which strand is to be copied, and the direction that is to be copied. Third, complementary bases are assembled, U instead of T. Next, a termination code indicates where transcription will stop. Lastly, the mRNA produced is called an mRNA transcript. So we have AUG, UUU, GGG, CUC, GUA, UGA. As you can see, the T's are replaced by the U's in the sense strand. Now, DNA is located in an organelle called the nucleus. Transcription and mRNA processing occur in the nucleus. The nucleus is surrounded by a double membrane after the mature mRNA transcript is produced. It moves out of the nucleus and into the cytoplasm through pores in the nuclear membrane. The next thing I'm going to talk about is transfer RNA triplets or anticodons. tRNA functions to transport amino acids to the ribosomes during protein synthesis. This is when transcription occurs. Translation occurs. The amino acids formed are tyrosine, lysine, proline, glutamate, histidine, and serine. Translation is the process where ribosomes synthesize proteins using the mature mRNA transcript produced during transcription. The ribosome attaches to mRNA and then moves along the mRNA, adding amino acids to the growing polypeptide chain. It includes an mRNA transcript, a ribosome, several tRNA molecules, and amino acids. A ribosome attaches to the mRNA transcript, and a tRNA molecule transports an amino acid to the ribosome. A second tRNA molecule bonds to the mRNA at the ribosome, and codes must match, and a bond is formed between the two amino acids. The ribosome moves along the mRNA to expose another codon for a tRNA molecule. Translation occurs in the cytoplasm, which is outside the nucleus. The process begins where the ribosome moves along an mRNA transcript and reaches an initiation code. The initiation code signals the start of a genetic message. Synthesis stops when the ribosome reaches a terminator codon. Termination codes signal the end of the genetic message. Bye! Thanks for meeting me here so that I can get my wings.